Without further ado, I would like to present to you the idea of today. And the idea of today is to bring brains to meet. So this is why we chose When Brains Meet, stolen from Yoram Cohen, the previous representative university in Europe. Present and future means present are the distinguished professors who do science today. Future are the potential scientists of tomorrow, the students of ELSC. So they come here and mix in time and topics. And I would like to invite Professor Bill Newsman from Stanford University, the Clark Center, and the BioX at Stanford, a member of the steering committee, and I will not make the official presentations, because if I start to talk about Bill today, this will take all the time of his lecture. So we are happy to have you here again, Bill, and thank you very much for coming.
I'm starting my timer. It's great to be back here. It's been about 6.5 years, I think, and it's marvelous to see the progress that has been made with the Safra Center. And I look forward to seeing what the next 5 years brings as well. So thank you for inviting me to serve on the advisory board. I'm pleased to be here. So I want to talk to you today about work in the lab on this topic of gating, a new look at gating, and about selective integration, how we determine which sensory inputs to integrate and make decisions on in a complicated environment. How do we accomplish selective integration? And I'm going to propose that it's through network dynamics, that there's a mechanism that has actually not been known before in neuroscience, not been considered. Several models have been considered for how selective gating might be accomplished, but I think this is a very general and powerful model, and I hope to convince you of that by the end of the talk. Most of the work that I'll show you is published already, but I am told that this is a complicated paper and very difficult to understand. The first time Richard Axel heard me talk about this, he said, "I didn't understand a word you said." And that's always a bad sign. It means I need to do a better job of explaining things. And so I hope that today we'll demystify this work a bit, and you'll come away with a very intuitive grasp of what we've done. I want to say that the work, the bulk, the intellectual heavy lifting here was done by Valerio Mante, a postdoc in my lab, who is now at the University of Zurich, and David Cicillo, who was a grad student with Larry Abbott, a postdoc in Krishna Shenoy's lab, and who did the really critical theory and modeling work that I'll tell you about. So an example of context-dependent decisions or flexible decisions is the famous Wisconsin card sorting task, where a patient or a human subject looks at a succession of cards, and in one context, the subject is asked to report what color he or she sees. So for example, the correct reports would be red, green, blue, yellow, red. But in another context or another instruction set, the subject reports not the color, but the shape they see. So circle, star, square, cross, etc. And there may be a third context where they report some different aspect. So this is what we mean by context-sensitive decision-making. And these kinds of context sensitivity is ubiquitous in our behavior, and we know very, very little about the actual mechanisms that underlie it. So Valerio and I started on this project about 5 or 6 years ago, really, training monkeys on a context-sensitive task. And we used the random dots, the moving random dots paradigm, with a twist. So these dot patterns that the monkey sees, one on each trial, the patterns have two properties. They have motion, so they can see whether the motion is drifting to the right or to the left, and then the dots have this property of color. And under one context, the monkey ignores the color, but he reports the direction of motion, and in the other context, he reports the color and ignores the direction of motion. So the context that the monkey's working in is actually signaled by the shape and the color of the fixation point, and which is yellow square in the motion context, and then a blue cross in the color context. Now, I'm just going to show you 2 quick videos. These are 750 milliseconds, so don't let your attention wander. You have less than a second, but these are pretty easy trials, but I just want to show you what these things look like. So right now, you're in the motion context. It's an easy trial, but you are to ignore the color and tell me what direction of motion you see, and that should be readily visible as left, exactly. Now here you just ignore the color, and I'm sorry, you ignore the motion, and you tell me the color that you see. What's the predominant color? Green. Okay, blue, somebody said blue. Okay, green. So yeah, this is exactly the task, and here's the task timing. The monkey begins a trial by fixating on a fixation point, and then two targets appear, one red and one green, and then the random dots appear for 750 milliseconds, and then there's a delay period where the animal has to remember the outcome of this choice, and then he receives a go signal, which is disappearance of the fixation point, and then he just flips his eyes from the center of the screen to one or the other target to tell us what he saw. If he's in the motion context, he moves his eyes to the right for rightward motion and moves his eyes to the left for leftward motion. If he's in the color context, he simply goes, he matches the color of the target. So he goes, if he sees mostly green here, the correct answer would be a saccade to the green target. And if he sees mostly red, the correct answer would be a saccade to the red target. And in the data that we'll talk about today, we're really going to concentrate on this period right here. I'm not going to show you any data from this period or from this period, only from the 750 milliseconds after the random dots come in and when the animal actually has to attend to the correct stimulus, integrate that stimulus over time, and reach a correct decision in order to receive a reward. Now, the animal actually sees one of 36 different stimuli across the course of an experiment. And it's just a 6x6 matrix where on one axis is the motion coherence. So this is strong rightward motion. This is weak rightward motion. And the negative signs mean weak leftward motion and strong leftward motion. So averaged across the columns here, you have the direction coherence. But averaged across the rows, you see that we have 6 different color coherences. This is strong red, this is strong green, and of course this is a continuum in between the red and green extremes. So the monkey sees each of these 36 stimuli in random order, and he completes 2 passes through this set, so 72 trials, before he changes context. So the contexts are coming in blocks. They're not varying from trial to trial. They're coming in blocks, and the change between the blocks is signaled. We flash lights, we beep a sound, we do all sorts of things. We try as much as possible to help the animal keep clear about what context he's in. Now, here's the behavior from one animal. This is averaged across, I don't know, several dozen sessions or something like that. And we're going to look at the behavior on the motion context trials, and then in a minute we'll look at the behavior on the color context trials. So here we're plotting the fraction of rightward choices, fraction of times the monkey reports seeing rightward motion, as a function of the motion strength from strong right to strong left. And you can see we have this nice pretty psychometric function, which means we're under behavioral control, and the animal really is ignoring the color and paying attention to the motion. But now realize we're going to play a trick right now, and I'm going to ask you to play this trick several times during the talk. Realize that every one of these motion stimuli also had a color associated with it, right? And we can take these exact same trials and we can resort them by the color of the stimulus. And when we do that, we can plot the proportion of the trials on which the monkey chose the green target as a function of color strength from strong green to strong red. And you can see 2 things here. First of all, this is basically a flat function, which confirms that the monkey is paying attention to the motion and ignoring the color. Secondly, though, you can see it's not perfectly flat, so the irrelevant stimulus is in fact bleeding through some. He tends to make green choices when you have a strong green stimulus and vice versa for red. And I can tell you through very thorough analysis that Valerio has done, when the irrelevant stimulus bleeds through, it's usually at these weak motion strengths here, which is not surprising. I think that's pretty intuitive. Okay, here's the other half of the trials, okay, when we're in the color context. And you simply see the reverse pattern of data here. And what this should convince you of is that the monkey can do this task fundamentally. He can work at threshold on 2 different tasks, generate nice psychometric data, and he can do this gating problem. So that means that somewhere in the brain, by some mechanism, there has to be a successful rerouting of information. Here, it's motion information that's influencing the decision process and ultimately governing the direction of the eye movement. And here, it's the color information that's doing that. So this is what we mean by flexible gating, routing of information flow in the brain. Now, when Valerio and I started this, we wanted to know about this gating process. We wanted to know about this switching mechanism. That's literally what we called it, the switching mechanism. We wanted to get physiological evidence for it. We wanted to measure its dynamics. Unfortunately, we did not get the results that we intended to. So the way we set this up, we know that there are circuits related to perceptual decisions in this task in the parietal lobe, LIP, in prefrontal cortex, and in the superior colliculus. So this is all work from my lab and Mike Shadlin's lab. We also know that the motion signals on this task come largely from extrastriate MT. I mean, we know that as well as we know anything in visual neuroscience, I think. Color is somewhat less, it's less secure because some of the critical experiments haven't been done, but almost certainly it comes from the ventral visual pathway V4 and IT. So our notion was that we have these streams of motion and color information coming into a decision process, and the decision process, of course, has to integrate the correct one and filter out somehow, ignore the incorrect one, and this is simply an integrator, and I say just literally a time integral of the relevant input, a la Shadlin, who has done, you know, tons of really beautiful modeling of this kind of diffusion to bound integration process over the years. And what Valeria and I expected to see was that in the motion context, MT would be hooked up to these decision circuits, but in the color context, MT would be out of the action, and it would be these other areas that are hooked up to the decision circuits. And we expected to see some kind of switching mechanism that we hope to actually measure at the single unit level. But that's not what we found. As you'll see, what we found is that motion and color reaches the PFC in particular. This is the only place that we have data right now, so I can't say for LIP or the superior colliculus. But motion and color reach the prefrontal cortex here in both contexts, leading to a bit of a conundrum about how the decision circuits know which one to integrate. It implies that gating or selection happens late. Now here's an overview of the rest of the talk, and I'm going to make one assertion here right at the beginning. I'm going to tell you that MT in this task appears to play no role in gating. So Valerio has a body of data where he did dual electrode recordings from MT and prefrontal cortex, and we were looking for changes in temporal coherence, as has been demonstrated in some other groups. We were also just looking for changes in firing rate. When the animal moves from one context to the other context, could we see anything different about MT responses or the way MT responses were temporally related to activity in prefrontal cortex? And the answer is no, we didn't see that, and that's a solid result in one animal. But what we did find, which I've already alluded to, is that in prefrontal cortex, so a lot of our recordings are from the frontal eye field, some from prefrontal cortex anterior to the frontal eye field, but here color and motion are completely intermixed at the single unit level, as I said, implying that gating occurs late, and I will document that for you with just one slide. The real bulk of the talk, I think the insight of this talk comes with a population analysis of the prefrontal activity, a particular kind of analysis where we look at the population activity in state space, and then we can see that color and motion are very nicely separable at the population level, even though it looks like a hopeless mixed up mess at the single unit level. And that's kind of going to be the first, that made a lot of difference to Valerio and myself, but there still remained the question about, even if they're separable, if they're mathematically separable signals, how does the gating actually get accomplished? And we still don't know that for the monkey cortex, but David Cicillo's neural network model is what gave us, for the first time really, a satisfactory and interesting way to think about this problem. So this is sort of the outline of the rest of the talk. Now, the very first step in analyzing the population data is actually to analyze neurons individually. So we recorded from individual neurons, and these are serial recordings. We are doing parallel simultaneous recording in the lab right now, but these were done serially. Sometimes we had 2 or 3 neurons on the electrode. Most of the time we only had 1. So we pretend like, in the population analysis, we pretend like they were recorded simultaneously. Now there are limitations with that, obviously, but there are a lot of things you can do. So for each neuron, we actually model the firing rate. We set up a model, a linear regression model for each neuron, and our independent regressors are things that we know, both from the animal's behavior and from previous results in prefrontal cortex and from looking at our own data. We know that these factors influence the firing rate of lots and lots of prefrontal cortex cells. So this is the choice. So we see decision-related signals up here. Kim and Shadlin published that back in 1999 for this area of cortex. We know that the motion coherency affects the responses of prefrontal neurons, as does the color coherency, and obviously these are incredibly important for the animal's behavior. And then we know that context if the animal's in the motion or the color context influences these prefrontal neurons and obviously influences the animal's behavior. For each cell, we included a bunch of interaction terms. Sometimes those interaction terms are significant, but not that often, and it's not where the main story is. The main story is really about dependency of frontal lobe firing rates on these factors here. Okay, now this is the only slide of single unit data that I'm going to show you. And what I'm plotting here are those beta coefficients. So these beta coefficients from the regression model. These beta coefficients capture how much choice is influencing the firing rate, how much motion coherency is influencing the firing rate, et cetera, et cetera. And right here, for a population of about 250 prefrontal neurons, we're plotting basically that beta coefficient for motion against the beta coefficient for choice. So each dot is a single neuron. And, you know, you can see that there are some cells that look like they're selective, right? If you looked at that cell right there, it has a large influence of the motion on the firing rate and very little influence of choice. It's near zero right there. And conversely, if you looked at that cell right there, you could learn a lot about the monkey's upcoming choice, but you wouldn't know anything about the motion coherence that led to the choice. So you can find cells that seem to be selective like that, but clearly that's not the story here. The story is that these signals are completely mixed with only a weak correlation between them. And the same is true if you look at the color beta coefficients against the choice beta coefficients, and if you look at the color against the motion. This is completely uncorrelated here. Now, yeah, go ahead.
Beta was time dependent, so
Yeah. Good question. So the question is, what happens to time here? We've got all of these betas that are time different. And the main thing we're using these beta coefficients for is to establish the direction in state space along which population activity varies. And what we really wanted to know is whether the beta basically points in the same direction. The norm, the amplitude of the beta changes from as we go through time. But we identified the beta, the direction of the beta, the actual direction in state space at the time where it's maximum. Okay, so when I show you these things, I'm identifying it where the motion signal is maximum, the choice signal is maximum, and the color. And those are different times for different ones of those things. But the direction of the vector is actually consistent through time, which we show in the paper, and I can go with you over that later. Okay. Now, for many of you, I mean, we all should have realized after 20 or 30 years of prefrontal physiology, right? This is a standard result in prefrontal cortex. This is what I used to call the prefrontal zoo, that you can find any animal you want to find in here, any combination of signals that you want to find. But we should all have realized a long time ago that this is actually an important finding because it means that color and motion at the population level are orthogonal. They're mathematically orthogonal to each other. Knowing something about the motion gives you how motion affects that cell, gives you no information about how color is going to affect the cell. And I've only seen one paper over the last 40 years that actually phrases it that way. It's a paper from John Duncan's lab published in PNAS in 2008, and they were headed down a very good direction with this and done more good work on it since then. So the message here is that these frontal eye field responses, color and motion, are completely intermixed at the single unit level. This is perplexing because we know that the monkey has to base his motion on color, his choices on color or motion. So how in the world does he do this if they're completely mixed at the single unit level? And so next I'll show you the results of the state space level that emphasizes this point of separability that we all should have tumbled to a long time ago, but at least we didn't in my lab. We were expecting something much simpler, simple-minded single unit physiologists. So I don't have time to go through the methods here. You can essentially, the methods are taking, if we have 250 neurons, you can regard the state of the system, the population, as living at some point in a 250 dimensional space that simply captures what the firing rate is of each of the 250 dimensions at any one point in time. And the essence of this analysis is to go through a dimensionality reduction exercise where we actually identify axes in the state space, axes of responses. You can think of them as patterns of responses across the population of cells that are related to the motion, to the color, to the context, and to the animal's choice. And the methods are published in this paper here, and I'd be happy to go over them with anyone who wants to talk with me about this later. But right now, I'm just going to give you a cartoon understanding, okay? So the cartoon is this. Imagine that we were recording from 400 neurons, sort of a 20 by 20 matrix of neurons. It doesn't have to be laid out flat like this. This could just be a unidimensional vector of firing rates. But each neuron has a particular firing rate, a high, medium, low firing rate like that. And the fundamental point of the dimensionality reduction is that we can identify underlying basis sets that when you add these things together according to certain weights, you actually reproduce the pattern of activity across the entire population. And what we can do is basically find a basis that represents choice, a pattern of activity among these 400 cartoon neurons that represents choice, a different pattern. Now, realize these two-dimensional patterns here mean nothing. I mean, they're just cartoons. They're not data, right? But fundamentally, it's a different pattern among the same 400 cells that represents the influence of motion at the population level, and then a different pattern that actually represents color. And if you add these three patterns together, each with a weight of 0.5, you actually reproduce this pattern here. So this is a way to identify patterns of activity in the population that relate to choice, motion, color, and of course there's another one for context. So what we actually do then, we look at the pattern of activity in the population, how it changes over time. So this one's different, right? There's the one I showed you first. This one's different. And what's different is that choice is dominating this pattern of activity much more than motion and color. So we can actually follow, at the population level, we can follow the influence of choice, motion, color, context by tracking these weights, all right? And that's the essence of the state space analysis. So what we have then is axes for choice that we have learned from the linear regression, and axes for color, axes for motion, and there's one for context that matters. And we plot then the population activity as trajectories in the state space. And this state space, I want to emphasize, many of you are familiar with this concept and are comfortable with it, but some of you are not. And I want to emphasize that there's nothing magical here. There's nothing mystical about it. All this is in the end is firing rates, okay? But it's patterns of firing rates across the population. I mean, if we were dealing in the original high dimensional space, every axis would be a firing rate. But with a reduced dimensionality, these axes here represent some ratio or pattern of firing rates across the population, okay? So, but it's not magic. It's all firing rates in the end. Now, this is an example of trajectories on average data for an experiment from one of the monkeys where here is a trajectory that was averaged across all the trials where the monkey made a leftward choice, and here is a trajectory averaged across all the trials where the monkey made a rightward choice. So a lot of things are buried in these averages, but the main thing I want you to see here is that when the monkey chooses left versus when he chooses right results in displacement along this choice axis, okay? And that's not surprising. That's the way we set it up. If our math is right and our data analysis is right, there should be displacement here. That's how we built this axis in the first place. But the thing that maybe is certainly surprising to us is that there is another signal that pushes these things off of the choice axis in these arcs. And this other signal is actually motion. It's what we call momentary motion because it's a motion signal that's actually orthogonal to the motion that's integrated into choice. I mean, choice depends on motion. If the monkey is in the motion context, right? He's in the motion context. So choice depends on the motion, but this is a component of motion that's actually orthogonal to the choice. So there is a separate signal here, not just the choice signal, but there's this sensory signal. And we can convince ourselves that this is motion simply by breaking this apart into the strong rightward motion, medium rightward motion, and weak, and similarly weak, medium, and strong leftward motion. And we can see that this signal is titrated according to the coherence of the motion. So this is a separate signal that we have here, and we refer to this as an axis of integration, where the motion signals are in the motion context, are integrated to reach a choice, and an axis of motion here that's orthogonal to choice. Now, I'm going to do this trick now that we did a while ago with the psychophysical data, and I'm going to remind you that everyone. Well, let's slow down, Bill. Let's be sure. Let's go through here with time. I have not explained the time dimension to you yet. Time is implicit in. Passage of time is implicit in these kinds of plots. So there's not a time axis. Time is passing from data point to data point. So this is the 750 milliseconds while the dots are on, and this is sort of the pattern of firing rates here when the dots turn on, and there's basically no difference here. There's differences between the context, but context not being plotted on here. So this is where they start, and this is the evolution of the population of activity as time passes. So every data point is at 50 milliseconds, and there will be 15 of those to get to 750 milliseconds. So this is the neutral state when the dots come on, and the population evolves here for leftward choices and evolves here for rightward choices. And I apologize for not explaining that at the beginning. Okay, so here we play this trick that we did in the psychophysical data, and we remind ourselves that all of these trials here that go into this average, all of these trials have a color coherence associated with them, right? So we can replot these kinds of things according to the color coherence of the stimulus, just ignoring the motion. And this is it. So the color coherence goes from cyan to dark blue, and you can see that when we replot these data according to the color coherence, it just looks like a mess. It looks like a complete mess. But that's because we're not looking along the correct axis. If we just rotate these data so that we're looking along the color axis, then you can see that there's this beautiful parametric representation of the color in the same population of neurons in the motion context, right? So this is not what we expected to see. We expected that the color would get filtered out before hitting the decision circuits, right? The decision circuits, neurons that predict the animal's decision accurately, they have to be responding to the motion in the motion context, not the color. And we thought this was going to be gone and would not be there. But we were dead wrong. And the color is there, and it's not modulated particularly by context. So if we go now and look in the color context, so this is the other half of the trials, and we plot by color, and we're looking at the color axis, you can see we have the movement along the choice axis for leftward choices and rightward choices, just as we did in the motion context, but now these arcs are related to the color. So this is strong color to weak color, indicating a leftward choice, and strong to weak, indicating a rightward choice. And if you replot these according to the motion, again, you get this mess. But if you just rotate the axes here, you see that buried in that mess is in fact a beautiful representation of the motion signal, even in the color context, right? So this really brings the problem to the nub of it. That here in this cortex that we have reason to believe is actually integrating signals toward a decision. I mean, we know that some of the cells are carrying these big choice signals. We have both the relevant and the irrelevant stimulus present. And the question is, how does one get selected to inform the behavior and the other get ignored? Yeah. Go ahead, David. Yeah.
You're really plotting here are the coefficients, beta for color, beta for motion, beta 1, beta 2, beta 3. This is just beta as a function of
Yeah, what this is, David, it's not single betas for single neurons. This is a collection of betas that defines the choice, right? If you have 250 cells and you have 250 beta coefficients for a choice, those 250 beta coefficients define a direction in state space. Okay, they just define an axis, and that's what we're plotting here. We're plotting that in that direction state space. All that is a ratio of responses from all 250 neurons. Now, we're not really doing this in high dimensional space. We've gone through a PC exercise first to denoise, but so this right here is it captures all of the betas for motion from all of the neurons. This captures all the betas from choice, and this captures all the betas from color. Is that clear? Yeah.
For color versus
yeah.
motion, and it appears that they're independent because there's no correlation.
Yeah.
It's just that you collapse over time dependence. So now the time dependence is here.
Yeah.
So in fact, this is a statement that they were correlated.
Okay.
But lost it with the maximum.
Sorry about that. Yeah. No, they're not correlated, David. We have to be clear what we're saying about correlation here. Let's go back and look at this. So what we're showing here, the directions in state space are related back to the betas calculated for the individual neurons, okay? But that betas, that establishes the choice, it doesn't say how strong that signal is, right? It establishes the direction in state space that corresponds to variability with choice, but that activity can wax and wane. So at the beginning of the trial, for example, the value of choice is near 0 because there is no choice yet. There's no information yet. In fact, there's no there's no motion, there's no color, there's no choice, there's no nothing. So it lies here near the origin. I mean, in this particular three-dimensional space, it would lie here near the origin. Now these axes are still defined, okay? But what we're seeing here now is the pattern of population changing, the pattern of the population response changing. And as it changes, is it moving along the choice direction in state space, or motion, or color, or all three? Yeah.
Would you expect a weaker choice axis dependence for the weaker signals? Here you've got a little bit of it on the right choice.
Yeah.
None of the others do.
Yeah, so these are asymmetric fundamentally due to the fact that all of these recordings were made in one hemisphere. So the eye movement is the output, and you know, if we're recording in prefrontal cortex, we get bigger responses when the monkey's making a contralateral eye movement than when he makes a ipsilateral eye movement. So we've got floors on this activity represented by low firing rates. That's why the asymmetry is here. I mean, if we recorded in the other hemisphere, it would be exactly the opposite.
The dependence on the strength of the signal.
Yeah.
For weak color, you would expect less choice.
Ah, right. Okay, good. Now I understand, Shaul. Okay, so Shaul was pointing out that if this thing were in fact acting like a perfect integrator, you would expect less excursion along the choice axis for weak, medium, and strong motion signals. And in fact, you see that here when he's making the eye movement ipsilateral to the receptive field, okay? When he's making the eye movement contralateral to the receptive field, you get this compression. And in Shadlin's integrator models, this compression here is related back to urgency of the animal. He can model this by adding an urgency signal, and we actually modeled that in our paper as well. But you are totally right. A perfect integrator, these things should be separated out here, and they should be separated substantially, actually. And they should be separated by more than this. And the animal's urgency to make a decision, knowing that he's only got 750 milliseconds to go, can reproduce these data. All right, so it's going to be a challenge here. I'm at 30 minutes. Okay, here we go. Okay, so we've done this. Let's keep going. Okay, so what we believe here is that we're dealing with these separable signals in state space, and what we have is a set of axes on the motion trials that are substantially orthogonal to each other for motion, color, and integrated evidence. And I have not shown you data from the context signal, but it's there and it's strong. And basically what the context signal does is just take this set of axes and it moves them to a different area of state space. Now that's not magic again. All that is firing rates. It just means that some cells have different firing rates when the monkey's operating under the motion instruction versus when he's operating under the color instruction. And anyone who's read the PFC literature knows that that's not a new observation from us. Earl Miller and John Duncan and many labs have shown these context-related signals. But what's interesting is that it just seems to be a displacement of this axis system to a different region of state space. It's not some fundamental change to the axis system. It's a displacement. In other words, the context signal itself is orthogonal to these signals. Okay, so interim conclusions here. Task-relevant variables are profoundly mixed at single neurons, but separate at the population level. Both relevant and irrelevant sensory inputs are present in each context. They are modulated very, very little by context, much to our surprise. So the irrelevant input is not filtered out prior to this integration step. And that means that selection must occur late, probably, we think, within PFC and related areas such as LIP and the superior colliculus. So they're separable at the population level. But the fact that they're separable still begs the question of how they get sorted out, and that's the subject of the final part of my talk, which is David Cicillo's modeling of these data. So Valerio and I were actually not happy after 3 years of recording work to get these results, and we just kind of had to wave our hands and say, well, it gets sorted out someplace else, maybe downstream, maybe within PFC, and we were going to write yet another hand-waving discussion section to another prefrontal cortex paper. But David Cicillo, when he came to Stanford, heard Valerio give this talk, and David said, my models will solve that problem. That's exactly what my recurrently connected neural network models are all about. It's learning dynamic problems like this, and I believe I can solve that. So he went to work on it in his spare time. And he and Valerio actually met each Friday morning for about a year, learning to talk each other's language. And David learning about neurons and Valerio trying to learn about these models and how they could actually do this rationally. And at the end, I think we got to a very good place. So David, as many of you know, did his PhD with Larry Abbott, working with recurrently connected neural network models, asymmetric recurrence. And so for those of you who are network people, this is a firing rate model. Okay, it's not biophysically realistic. Each individual unit is nonlinear. It's a tan H compression on the firing rates for strong inputs. And importantly, these have asymmetric connections. So if A is connected to B with a weight of 0.3, B is not connected back to A with 0.3. It can be anything it wants to be at the end of the training period. Initially, they're all initialized randomly. And that gets us into the world of non-normal matrices and non-normal dynamics in networks that have particular matrices like this. And that's actually going to be important to the story. At the beginning, all of these weights are initialized randomly, round zero, so they're both positive weights and negative weights. So this is the engine. There are 100 of these units here. I will slip and call them neurons. I should say units. On every trial that we simulate, the motion signal comes in, a noisy motion signal as a function of time, comes in and is, again, with randomly selected weights connected to all of the 100 internal units, and simultaneously a noisy color input comes in. So this is exactly what happens with a monkey, right? And the motion signal, it can be offset to higher levels for strong rightward motion and lower levels for strong leftward motion. And similarly, color can be offset for red or green. So these are just simulations of the inputs. The other important input, of course, to the network is context. So the network, like the monkey, has to know what its instructions are. And the context is usually, it's a 0 and 1 toggle for the motion context, and it's 0 and 1 here for the color context. So it's just a binary toggle that indicates the correct context. And then we use this neuron here, a linear sort of sum of these weights onto a single neuron out here as a trick for reading out the internal integration state of the network and in a sense, rewarding the network. It's a back propagation style learning rule that David Cicillo worked out in his graduate work. So what the network is trained to do, given the combination of context and sensory input, the network is trained to integrate the correct one that's signaled by context, to integrate the correct input and ignore the other one, toward a +1 if the correct choice is rightward, and toward a -1 if the correct choice is leftward. Okay? And this is really interesting because this is just a generic vanilla recurrent architecture. We are not baking a solution in here. We are asking the network to see if it can come up with some way to solve this problem. And then what we want to do is go in and reverse engineer the network and learn what it has learned. So David now uses training methods from these two guys up in Toronto. He says they're better than the ones that he invented as a graduate student, and it's the training methods from Sutskever and Martins that he actually uses now. So this may be 2,000 repetitions before the network actually learns this problem. And then you lock in these matrices of connection weights, right? We know the complete connectome here. We know the connectome of these inputs and the toggle outputs. And this should convince you that the model can learn the task. So this is behavioral data in motion context, choices as a function of coherence, and scrambling these and looking at the color strength and the motion context. And we basically reproduce what we saw in the monkey psychophysics, right? So I just show you this to convince you that the model actually learns to solve this task. Okay, so he can then, once the model learns the task, David can then look at the model using the same kinds of techniques that Valerio looked at the PFC physiology. So just treat these as units and do a state space analysis on their activity, derive these choice axis and the color axis and the motion axis the same way that we did, and look how the population representation in the network evolves as a function of time, where time starts here at the onset of the visual stimulus and then goes out here to the offset of each of the stimuli. Now, Shaul, you notice here, we're really acting like a perfect integrator. I mean, this network really is a perfect integrator at the end. So the termination points are actually substantially different, which they're not here. I'm just reproducing the physiology here to show you guys this. And so it's acting as a perfect integrator. There's some things that are very similar between the model and the physiology. And one of the things that's similar is that if you resort these by the motion, again, just as in the physiology, it looks like a mess that there is no motion signal. But if you simply rotate so that you look at the system along the motion axis, In the model, just as in the physiology, you see that there is a beautiful representation of the motion signal, just as there is a beautiful representation of the color signal. They simply have nothing to do with each other. But both signals are present, okay? And that is really interesting. There's some things that are different about the model. I mean, one of the most, the biggest difference you see here is that the model keeps integrating right out until the end of the 750 milliseconds, whereas the prefrontal cortex doesn't. It seems to integrate for a while, and it has this motion signal for a while, but then these trajectories dive back to the choice axis, basically, and it looks like this motion or color signal disappears. And we think that's because, and we have independent evidence, that the monkey is basically making up his mind after 400 milliseconds or so and is just ignoring anything that comes in after that. So we think this is because he's quick on the trigger. And of course, we could mimic that by shutting off these inputs to the model, but we didn't want to mimic that. We just wanted to see, I wanted you to see what's actually happening here with as few bells and whistles as possible. Okay, so the model develops you can describe its activity in this four-dimensional space, just like in the physiology. But here's where David's contribution really came, and this is where the mechanistic insight comes. The question is, how does the model actually do this? And so what David did then was reverse engineer the trained model using methods that he developed with Omri Barak at Columbia, and basically this reverse engineering comes in several steps. The first point is in the trained network to identify stable fixed points or attractors. There are none in the untrained network, by the way. Then around each fixed point, simply doing a Taylor series expansion to estimate a local linear dynamical system that approximates the local dynamics around each fixed point. And remember this connection matrix around each fixed point, the approximation is going to be non-normal. And then, since this is a non-normal connection matrix, there's you can identify both the right and left eigenvectors associated with the mode of slowest dynamics. So these are near zero eigenvalues. In a normal matrix, you would just have one of those, but in a non-normal matrix, you have two of those, and that's going to be important. For those of you who don't understand any word I just said on this slide, don't worry about it. It's not a big deal. I'll explain to you what's happening. But these two eigenvectors, the right and left zero eigenvectors, capture two emergent dynamical features. And these two emergent dynamical features explain integration of evidence and the context-sensitive selection in the network. So it's these emergent features that are really critical. So here's the model response in the state space, and these are just, again, the state space is just patterns of firing rates among the model units. And here's an example of the fixed points that David found. So you can find these fixed points. He can get these local linear systems around the fixed points, identify the mode of slowest variation, and they all line up with each other. Amazingly, they point in the same direction, which means that they approximate a line attractor. And a line attractor is exactly what you want for memory, right? The line attractor takes inputs. It responds dynamically to its inputs. And once the input disappears, it holds on to it. The system stays in that position that reflects the inputs that it has gathered. So this approximate line attractor is one of the emergent features. It's one of those near zero eigenvectors. It's either the right or the left. I actually can't remember right now. I think it's the right zero eigenvector. Heim would know, I'm sure, but I'm not sure. It doesn't really matter which one it is. But that defines the line attractor, okay? Now, if we blow in and look at one of these attractor points, now David has the advantage of being able to do these beautiful perturbation experiments. Any engineer or applied physicist would say, well, how do you study a nonlinear dynamical system? You do it by perturbing and then watching the system relax. So David can put the firing rates at any place he wants. So he can take these 100 units and stick them right here on that line attractor, and they're nice and stable. They stay there. That's what an attractor is all about. But then he can put in a pulse of activity, say in the color direction, in the color context, and he puts in a pulse of activity along this color axis, and he watches the activity relax. So this is a population of units. They're changing their firing rates. They come back to the attractor. That's what systems do. They come back to points of attraction, but it comes back to a different location on the attractor. And if you put in another pulse, you come back to a new location on the attractor, and you keep doing this. If you just go pulse of red, 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 you just keep marching out along the line attractor. In other words, the system is remembering how much red evidence it saw. And if you did green, you would simply go like this, and you would march out in a line attractor toward a leftward choice like this. But what happens now to the irrelevant input? So he can pulse into motion, right? So he pulses into motion and he watches relax, and it relaxes back to the line attractor to exactly the same place where it began. And you can do that over and over and over again, and it comes back to the place where it began, okay? So this is really critical. The dynamics here that you measure with these perturbation experiments, the dynamics incorporate integration in the relevant dimension, but they simply pay no attention to the input in the irrelevant dimension, okay? And so the secret here to selection is in the relaxation dynamics that are caused by this network of connection weights that have been learned. So here's a summary of where we are. We're living in a space now where there's this line attractor that corresponds to the choice that the animal makes. There is a direction that corresponds to color input, a direction in the space that corresponds to motion input, and these are independent of context. So if I plotted the context up here for you, it would be going in and out of the board, okay? It would be orthogonal to this system here. So these are independent of context, and you say, well, how is the context happening? It's happening due to the relaxation, which is another word in dynamical systems for flow fields. So it's really, the secret is in the flow fields. And the flow field, the direction of the flow field indicated by these arrows is that other zero eigenvector, okay? It's the second emergent feature. And this flow field is slanted like this, and it always lines up with the relevant inputs. It lines up orthogonal to the relevant input. So as you change from the motion context, you watch these arrows on the flow field, you change from the motion context to the color context, and this flow field changes orthogonally to each other. Now how does...
Three-dimensional, right? How can you sketch it in the three-dimensional?
Yeah.
It's not the line, right?
Yeah. Well, the third dimension here, I mean, yes, I can't express all the dimensions in the two-dimensional figure. That's right. Yeah. So look, I'm almost finished here. Let's work this out. Let's work it through carefully in this example. So let's say we're in the color context. We start here, but the system experiences a color input. So the color input displaces you along the color axis to a new point in space. And of course, that's just ratios of firing rates. That's all it is, according to the color pattern. But then the system will relax, and it relaxes along the direction of these flow fields. So here, the color is getting integrated in the color context, right? But if you put a motion input in, it will also relax back to the line attractor. But again, it conforms, its direction of relaxation conforms to the direction of the flow field, the second emergent feature of this connection matrix, and it simply is not integrated. So it's the same dynamical mechanism that does integration and selection, and that's really important, okay? It's the same pattern of dynamics here that's responsible for integrating the relevant input and for ignoring the irrelevant input, okay? And it's a new way to do selection that, as far as I know, in neuroscience has never been thought about or never been anticipated. So the conclusions here is that gating is not about suppressing the irrelevant input, but it's about selecting the relevant input in state space. Gating is just one aspect of a dynamical process that occurs in the same circuit in the model. It occurs in the same circuit as the integration of evidence. It's all one piece of the dynamical system. And a final thing I want to emphasize, and I know that I'm in a friendly audience here to a large extent. I mean, many of these concepts have been applied in neuroscience for the first time here, but this, I assure you, there are many audiences, neuroscience audiences, for which this is not a friendly statement. And the statement is these dynamical constructs, which I think are the real explanatory power, where the parsimony and the predictive power lie, attractors, etc. These are inherently population concepts. We can study single neurons with PSDHs until the cows come home, and we will never learn things about attractors and about flow fields following perturbation. So I think this is an extraordinarily important thing. I don't know that our model is right or wrong. It hasn't been tested in real animals yet, but it's at least novel and interesting and has a way to predict things that are very difficult to predict from connectomes, for example. So it has interesting implications, and I invite questions about this afterwards. It's a general mechanism for gating. It scales up really well to multiple inputs, whereas Boolean logic does not scale up as you get multiple inputs. This one scales up nicely. It's easy to wire, right? These wiring on these networks is initially random, and I think we're going to hear from Richard Axel that there's a lot of evidence for initial random wiring in olfactory cortex. And another thing that I invite questions about is that there are many solutions here. There are many connectomes that will actually yield these two emergent features. And so it's very robust. The synapses don't have to change their weights very much to find a solution. There are issues. Number one, this training procedure is not at all biological. That's a real challenge. I understand this is why these asymmetric networks have been ignored for 30 or 40 years, basically, because they're so difficult to train, and the methods that are currently used are not biological, and that remains a challenge. Some people have said, well, your random asymmetric wiring doesn't look like biology to me. You know, it doesn't look like the cortex. I mean, I personally think that the overwhelming likelihood is that the connections between any two cells in cortex, A, B, are likely to be asymmetric. And it doesn't really matter whether they're lined up in layers for the purposes of this model. But I'm betting that asymmetric wiring, which is the real heart of this mechanism, is going to look biological. But you know, this is the key thing, right? How do you test such a model? And of course, the way you want to test it is through perturbation relax, just like David did his model. And this is something that's not widely appreciated. This would be, optogenetic tools would be ideal for this, but not the current optogenetic tools. Current optogenetic tools may just hit all the layer 5 cells, or all the layer 3 cells, or all the inhibitory cells. But the patterns of firing rates here are not just layer 5 cells, and they're not just inhibitory cells. You want to actually be able to reach out and touch these cells individually and reproduce the actual patterns of firing rate you see during behavior. And you want to be able to displace the system to any arbitrary point in state space and watch it relax. And it's in that relaxation that's the acid test of the model, right? Do you see the line attractor? Does it relax to predictable points along a line in state space? And is it asymmetric for relevant and irrelevant inputs? And that's going to take what Rafi used to call playing the piano style of optogenetics. And realize that's a use of optogenetics not for changing behavior, right? Most of the time, we try to change behavior, or we try to change the responses of cells with optogenetics. This is a different use of optogenetics. This is to perturb a system and watch the internal dynamics of the system. And that's a new way, I think, to think about optogenetics. So I will stop here, and sorry, Elon. I mean, I went too long, and I understand if there's no time for questions, and maybe we should go on to the second.
I don't think we can let this go without any questions, so please, yeah. Shaul?
What about the trials where the monkey makes a mistake?
Yes, good question.
For the weak signals, there are lots of them, 30 or 40%.
Yeah.
Where does it diverge from the trajectory?
Yeah. What you see is what you see. The question is that in the trajectories that I saw Shaul very sharply noticed that on the plots I was showing you data from correct trials only, and he's saying, what about error trials? How do things look in error trials? And it's a great question, and I'm trying to put one of these up here, for example. I don't have the actual data, but I can describe this to you. What happens in error trials is that it looks like the correct trial, except it goes with the animal's choice. So if the animal chose leftward by mistake, then the trajectory goes along here as a leftward choice, and its displacement in the motion direction is weaker than for the identical motion signal where the monkey got it correct. So it's as though it behaves the same way. The data predict the animal's choice, but it acts as though this motion evidence is weaker. Yeah. Udi.
Regarding timing, I think this slide is very relevant, and also in terms of the space. I expect that when you have strong motion, the monkey reaches a decision earlier than when you have weak motion. Now, you mentioned that typically the motion would integrate over just 400 milliseconds and stop.
Yeah.
But if it's very weak, it would take more time.
Yeah.
And I would expect that with strong motion, following Shaul's point,
Yeah.
the trajectory should be faster towards the choice left
Okay.
than it would for the weak motion. But I don't see this in this graph, and I was wondering also, well, let's stick to that.
Yeah, I agree with you. I don't see it either, Udi. And I understand the question, which is that the dynamics ought to be different. One might expect the dynamics to be different on strong motion trials as opposed to medium or weak motion trials. And you might expect to see more rapid accumulation. If we actually project these data onto the choice axis, there is evidence for more rapid integration on the strong motion as opposed to the weak motion. But I think Udi was also saying that he might cut that off. Once he feels like he has enough evidence for the decision, he might cut that off, and this return to the choice axis might be faster as well. And as you say, I mean, there's not a lot of evidence for that here, and I don't know. Sorry?
There are less time points in the stronger ones.
There should be the same numbers of time points. Yeah, yeah, they're just closer together here. Yeah, I mean it's a great question, Udi, and I don't know the answer. There's one question in the back. One last question. Okay.
The simple binary choice is a pretty arbitrary kind of experimental situation.
Yeah.
So how would you suggest an extension of these kind of models, especially that line effect, to something where you actually have a full set of possible behaviors?
Yeah, I think it's a really interesting question. I mean, we'd have to define the behavior first. So one example of a behavior might be that the animal doesn't just have binary context, that he's actually forced to integrate the motion and color. So if it's leftward motion and red color, he does one thing. If it's rightward motion and green color, he does another thing. And David speculates that in those situations, the model might learn to implement a plane attractor. And it would be interesting to see if we ever got evidence for something like that in the actual animal. So it's a matter of more choices and a matter of more sophisticated context, I think, that would get us up above this simple system. Yeah. Okay. Thank you.
I hate to say this, but I remember a long time ago that I visited John Hopkins when I went for postdoc. The big professor said, wow, this is an example of work that we want to do, an example of the way to present it, of superb study and superb presentation. Thank you very much. And I think we stay in the kind of the same spirit of combining theory and experiment, and this is the spirit of today. And here I'm involved myself, since Ben is my student, and David, and is a student of David, and they found each other at the center, in the coffee corner that we plan to have in the new building. And after several years of working in the lab, in the experiments, they continue to do some modeling work together. So Ben Engelhard and Ran Bershan will present their project, Physiological and Computational Mechanisms of Behavioral Learning.
Thank you very much. It's a pleasure to be here. This is the last talk of my PhD, so this is a wonderful setting to give it in, and I appreciate the opportunity. So I will present work done in the lab of Aaron Vadia, where we tackled the question of learning. And during my PhD, I worked with brain-machine interfaces, and when people usually hear the phrase brain-machine interface, or BMI, they think of a neuroprosthetic device that's designed to enable subjects that are paralyzed or have motor deficits to control an object in the external world, such as a robotic arm. But this is not what I did. Very early on, I decided that I want to use this as a tool to study the brain. And the way we chose to use this tool is in a very simple paradigm. We call it neural operant conditioning, or it's sometimes called neurofeedback or biofeedback. And we simply ask the brain to produce a specific pattern of activity. And in order to enable subjects to do that, we give them feedback about the neural activity, because we don't have access to the internal workings of our brain. But if you give subjects feedback about the neural activity, they're actually able to produce a very, very distinctive types of brain activity. So I'm going to argue that this tool is useful to study especially learning in this talk. So this was a paradigm that was made popular by Eb Fetz back in 1969. So here you see his work. He taught monkeys to increase the firing rate of single neurons in prefrontal cortex, in motor areas. And that was really the first demonstration that monkeys have the ability, subjects have the ability to precisely control activity patterns using feedback. And we used this work, this was published last year, where we actually taught monkeys to produce gamma oscillations in the local field potential. So these are oscillations that people have been researching for many years. They have been related to many cognitive tasks, and they have been implicated in brain disorders. but the role in the computation, in the network dynamics hasn't been clear, especially with regards to single unit activity and synchrony. So this is an example of our work. Here you see very nice oscillations produced by the monkeys after giving them rewards for it. This is raw data, so it hasn't been bandpassed in any way. You can see the oscillations by eye. You don't need to do any fancy statistics. And when we record single unit activity, you see that the spikes, which are plotted on the LFPs, are clustered in the falling flank of the LFPs. So we found that there is strong phase locking, and this phase locking implies very precise time neuronal synchrony, and we can quantify how the amplitude of the oscillations related to neuronal synchrony. So if you're interested in precise time synchrony and gamma oscillations, you can read this paper, but in this talk I will talk solely about learning. And this is why I think this is a great paradigm to study learning. So this is basically the main idea behind this whole talk, and this is something that we had early on. And we want to study behavioral learning. So how do typically people study behavioral learning? They have an animal or a subject, and they give him a task. So in a typical task, we know what is the desired behavior. We know that the monkey should push this button or the other button. It should do some kind of reaching movement. But we really have no idea what the neural activity should look like. So we don't know what is the desired goal of any type of network in the brain. And in contrast, in a neural control task, in a neural feedback task, we know exactly what the network should do because we are determining this task. So we know, for example, that the network should increase the firing rate of one neuron. And I think that this key difference enables us to do the data analysis and the computational modeling, which I will show you, to understand this type of learning, which we couldn't do if we use a normal behavioral learning paradigm. So this is our setup. We use the famous Utah array, which most of you are probably familiar with. This is a 100 electrode array implanted in our case, in the arm and hand area of primary motor cortex. And here you can see nice signals that we get. These are single unit spikes. We get these signals mostly at the beginning of the experiment after implantation. They actually die out pretty fast, but if the implantation was successful, at least at the beginning, we get nice recordings. So we have a limited amount of time in which we can do these experiments and get nice single units. So this is what it looks like. We actually chose to replicate the basic FETS experiment for this task. And here is our data. You can see that we asked monkeys to increase the firing rate of a single neuron. I will explain what these colors mean later, but this is the part of the session when the monkey has to increase the firing rate of this neuron, and you can see that he managed to do it very well. And the feedback he receives is very, very simple. Here you see a cursor in yellow and red. I know it looks strange, but it is a cursor, and here you see a target on the green, and the monkey already been trained in a movement task, so he knows that when the cursor reaches the target, he gets a reward, and this is something that he wants to do, and in this case, the position of the cursor is determined by the firing rate of this one neuron. So in a 400 millisecond window, we count in spikes. If the firing rate is high, then the cursor moves towards the target. If it's high enough, the cursor reaches the target, the monkey gets reward, and this is why he knows he has to do this over and over again. And we keep making it more difficult, so he has to increase the firing rate, as you can see here. So here's the main idea. We're using a very small perturbation, which is the conditioned activity of one neuron, and at the same time, we're recording a lot of other neurons in the local network, in the motor cortex. And we don't have any other constraints. The network only has to increase the activity of one neuron. And this is how we try to see what is the network doing, how does it learn, how do the other neurons change, and we can also relate the observed dynamics to different states, behavioral states, for example, a movement state versus a non-movement state. So this is what we decided to do. Now, there is a very basic question that we need to answer before we approach the question of learning per se. And the question is, how is neural control accomplished? This is a basic question in the field. There have been debates about it. This is, and one prominent hypothesis is that animals and humans presumably use neural control by basically imagining or planning movements. So this is a paper by the group of Richard Anderson in Caltech, and this was published last year. They showed they did the same type of task in parietal cortex, and they showed that when first they did the monkey doing movements, they had to plan movements, and after that they had to increase the firing rate of a single neuron. And what they showed is that during neural control, the activity of the other neurons appeared to be coding for movements. So their conclusion was that the monkey was doing planning or imagining of movements, and maybe that's how neural control is accomplished. And in that case, this wouldn't be a very interesting paradigm to study learning, because we could merely study movement-related patterns. So the way they set up their task is that first they have a movement block, and immediately after, they had this neural control block. So when I saw this paper, I thought that it could be that when the learning started here, the context of the animals was the context of doing movement. So, no, I don't want to do any updates. Thank you. I will postpone it. So I thought that maybe the context of learning was the context of movements, and maybe that's why they got the results. So maybe if the context of learning was different, then we wouldn't get the same result. We wouldn't see that movement neural control is dependent on movement-related patterns. So see, here's how we set up our task. We have first a movement block where the monkey is doing gripping and reaching movements, center out movements to different points in space. And then we have a block we call the pseudo-conditioning block, in which the monkey's both hands are restrained. He's looking at the screen. There is a cursor moving towards different targets, but the position of the cursor is random. And so the rewards he's getting is random and well. So basically the monkey is just sitting here with hands restrained, getting rewards and not doing anything. And after that, we have the neural control block in which the monkey has to increase the firing rate of one neuron, and everything else is the same as the pseudo block. So the monkey's hands are restrained, is watching the screen, but now the position of the cursor depends on the firing rate of this one neuron. And after that, we had a last movement block for control. So if our hypothesis is correct, if the network learns by adapting activity patterns from the pre-learning state, then we would see that the activity patterns during neural control would resemble more activity patterns adapted from the pseudo block and not movement-related patterns. So this was the hypothesis that we started. And basically, when people think about computational models of learning, this is really what most people do, because you think about learning as the network starts from a given state, a given neural state, and then it slowly adapts it in order to achieve the desired outcome. So we thought that this makes sense as well. So first of all, we want to establish, to test this hypothesis directly, whether actually there are movement-related patterns during neural control or not. Does the monkey imagine different movements in order to achieve and to accomplish the task? So here's why we decided to test it. We assume that if there is coherent movement coding across the population, then if we divide all the unconditioned neurons into two halves randomly, then each half should code for the same direction, because all the population is coded for this one direction. So if we divide it, each half should code for the same direction as well. And so here what you see is a matrix of the probability of joint coding of the two halves. So this is the targets coded by one half of the population, and obviously we randomly divide it and the average many times. And this is the targets coded by the other half of the population. And if there is coherent coding, then both halves of the population code for the same target. And then we would expect strong values along this main diagonal. And indeed, when we do this for the movement block, we get very strong values along the main diagonal. So we have very strong coherent coding during movements, which is not surprising because we are recording from primary motor cortex. And you see that the probability of joint coding is very high. And this is example for one session. But when we look at the same session during neural control, we see no evidence for that. There are no strong values along the main diagonal. And in fact, when we sum up the main diagonal, the probability is 0.123, which is very close to 0.125, which is 1 over 8, the number of targets. So this is basically chance level. Because by chance level, if one half is appearing to code randomly for one direction, and the other half is appearing to code randomly for the other direction, then chance level would be 1 over 8. And when we summarize these results for all the sessions, we see that in none of the neural control sessions, we had coherent coding above chance level, but we had this coherent coding in all the movement sessions except for one. So this tells us that it doesn't appear that the monkey is coding for movement in this task. The monkey is not accomplishing the neural control task by imagining movement, but maybe something else is going on. Perhaps our hypothesis that he's adapting neural patterns from the pre-learning state is correct. So now we want to test to see what actually happens during learning. And the first thing one does is to look at changes in firing rates. So now I'm going to plot the changes in fire rates of all the unconditioned neurons from the pre-learning, from the pseudo block to the neural control block. And this is what we see. We use a normalized index to compare neurons that have different overall firing rates. So this is an index from -1 to 1, where positive numbers mean that the firing rate increased during learning, and negative numbers mean that the firing decreased during learning. And what we see here is something very interesting. First of all, we see that in orange, these are the significant changes, and in gray, the non-significant changes. The red are just the condition unit for comparison. And what we see is that most of the units, about 2/3, significantly change the firing rate. So we saw large-scale changes across the population. But the next interesting thing is that these changes are relatively balanced across decreases and decreases. The number of decreases and decreases is not significantly different from an even split. And this was very surprising to us because all we asked the network to do is to increase the firing rate of one neuron. And you can imagine that one possible strategy for the monkeys would be to increase the firing of all the population, right? The monkey goes wild, all the motor cortex increases its firing, and they accomplish the tasks. And in fact, this would be a good strategy for the monkey because each day, each session, we changes the conditioned neuron we chose it randomly from a different electrode. And then the monkey had to learn, presumably, which neuron it was to increase its firing rate. But if the monkey had this global strategy of just increasing the firing rate across the population, then it wouldn't have to learn anything, and it would accomplish the task each session without working too hard. So, but this is not what the monkeys did. The monkeys actually had symmetric changes between increases and decreases in fire rate. So this gives us one clue about what is going on in the network during learning. So now we're going to ask the question is actually what is the nature of these changes? Why do some neurons increase their firing rate? Why do other neurons decrease their firing rate? And if our hypothesis is correct that actually learning involves adaptation of the neural patterns from the pre-learning state, then we should find some kind of relationship between activity patterns in the pre-learning state and these firing changes that we see here. And so this is the next analysis that we did, and actually we could only do this analysis because we have this neural control paradigm, because we know what is the important feature of learning. We know that the important feature of learning is the firing rate of this one conditioned neuron. So what I did next is I grouped all the unconditioned neurons depending on the sign of their correlation with the conditioned neuron before the learning. Before the monkey even knew which conditioned neuron it was, because we didn't even choose it. So here on the right are neurons which were positively correlated with the conditioned neurons before learning, and on the left are neurons which were negatively correlated with the conditioned neuron before learning. And what we see here is that neurons which were positively correlated with the conditioned neuron strongly increase their firing rate on average. And neurons which were negatively correlated decrease their firing rate on average. And as you see, this is a very strong result. So this actually tells us something very significant about how the network accomplishes learning. There is a strong relationship between the correlation patterns before the learning and the firing rate changes of the neurons. And we can actually use that to predict if a neuron will increase or decrease its firing rate depending on what is the sign of its correlation with the conditioned neuron before learning even starts. And when we do the same analysis for the movement block, we don't see any type of relationship, so the correlations during movements do not predict if a neuron will decrease its firing or not, but only the correlations during the pseudo-conditioning block, our pre-learning block. So this is a state-dependent effect. It depends on the state that we're looking at. It's not that there is an overall correlation that will predict these firing rate changes, but in fact, it only works if we look at the correlation pattern in the pre-learning block, and this is another example of why we don't think that this has to do with movement-related activity. So up till now, we've looked at changes in firing rates, and now we will look at changes in the strength of the correlation. So in order to do that, again, we have a normalized index, where positive numbers means that the correlations increased its strength. So the amplitude of the correlation increased during learning compared to the pre-learning state, and a negative number means that the correlation were decreased in strength. And here I'm plotting on top the correlation with the conditioned neuron. And what you can see is that most correlation became stronger. So correlations of all the unconditioned neuron with the conditioned neuron became stronger during learning. And when we do the same analysis between pairs of unconditioned units themselves, we see a different thing. So here we see a lot of changes again, but these changes, similar to the firing rates, are relatively balanced between increases in strength of correlation and decreases in the strength of correlation. So there are, again, wide-scale changes, but these changes are relatively symmetric. So this gives us another clue about what's going on in the network. So the first thing that one would say when looking at that is say, well, there are wide-scale changes across the populations, or something is happening during learning. But when I saw this graph, I thought that maybe I'm missing something. Maybe this looks like sort of a symmetric mess because we are lumping all these pairs together. But what would happen if we actually separated the population into different functional groups? Maybe we actually see a pattern in the changes of correlation which would make sense to tell us a little bit more about what happens during learning. So what we did next is to separate these neurons into three groups. Here are neurons which increase their firing rate significantly during learning. These are neurons which decrease their firing rate, and these are neurons which did not significantly change their firing rate. And now I'm going to plot the changes in the correlation strength for both positive and negative correlation between and within these groups. And when we do that, we get the following pattern. So positive correlations are in red, negative correlations are in blue. A wide line means that the correlation became stronger. A thin line means that they became weaker. And a dashed line means that they did not significantly change. And what we see here is actually a very interesting pattern that makes sense in terms of the changes of correlation. So between neurons that increase their firing rate, the positive correlation became stronger, but the negative correlation did not change. Something very different happens between neurons that decrease their firing rate. The negative correlation became weaker, but the positive correlations did not change. So these two groups these two functional groups of neurons actually are becoming more similar to each other in very different ways. And between these groups, both things happened. So the negative correlations became stronger, and the positive correlations became weaker. So these two groups are becoming increasingly anti-correlated, which makes sense because they have sort of have an opposing role during the neural control process. And there was also something very interesting that happened in these groups of neurons. So these are neurons that did not significantly change the firing rate. So when you look at these neurons, the first thing that you would think is that, well, these neurons do not really participate in the task because they're not changing their firing rate. But when we look at the correlation between these neurons, we actually see that both the positive and the negative correlations became weaker. And in fact, this was the most statistically significant result of all this graph. So this provides us evidence that these neurons which appear not to participate in the task actually became strongly desynchronized during learning. And so first of all, this means that these neurons actually did participate in the task, at least they were affected by the task. And the second thing that we thought about is, well, what would be the role of such desynchronization? And we're still speculating about that, but we have one idea why this would actually make sense during the task. So you can think that, well, these neurons participate in tasks. They're actually supporting the learning process by increasing the firing rate. Presumably the conditioned neuron is one of these. And this is neurons that are decreasing the task. And if they're becoming anti-correlated with the conditioned neuron, then maybe they're helping it increase the firing rate as well. But these neurons are not changing the firing rate at all. So maybe they're just representing a noise element in the system. And if in fact these neurons are basically adding noise to the system during learning, then the fact that they're becoming desynchronized would mean that their discharges are affecting less the other neurons in the network. You can think that if the spikes are aligned, then they would have more effect upon other neurons in the network. If the spikes are not aligned in time or not correlated in time, they would have a weaker effect. So maybe this is actually an interesting mechanism for decreasing noise during learning, but obviously this is a speculation. We don't have further data to support that hypothesis. So we saw all these interesting changes. We're actually getting sort of an inside look of how the brain learns, how the local network learns, but we really want to understand what is the computational nature of these results. So what kind of computational model could explain what we saw right here? And the first clue we had is, again, we saw very nice symmetric changes in firing rates and in correlations. And so this means that there should be some kind of homeostatic mechanism in the network during learning that maintains these changes to be relatively symmetric. And when I saw that, I thought that maybe a network that balances excitation and inhibition could actually explain this type of results, because we know that in this network, because of the balance between excitation and inhibition, if a group of neurons would increase their firing rate, then under some condition we expect that another group of neurons should decrease their firing rates to maintain the balance. And this argument is actually also valid for the changes in correlation that we saw. So when I saw that, I went to talk to Ran Darshan, whom I know, and he worked with David Hansel on these balanced network models, and we started a nice collaboration to try to develop a model to explain all these changes. And this is the model we came up with. I will not talk about it too much. Ran will talk in a few minutes. He will explain it much better. But the main idea is that motor cortex is modeled as a balanced recurrent network. This is a chaotic balanced network. And this explains the symmetric changes in firing rates. And then to explain the volitional nature of the neural control task, we have the volitional, what we call the volitional command or the volitional signal that's coming on from outside the motor cortex through feed-forward projections. And this is actually not a very controversial idea. We know that, for example, when we do reaching movements, then there is increased drive to motor cortex. And this is basically the same idea, except instead of doing a reaching movement, we're doing a neural control task, but it's still an action that's being performed in motor cortex. And so, and also we're only changing these weights. These feed-forward weights are the only one being changed with a simple reinforcement learning rule. So, does this model that has excitation and inhibition, it has a balanced excitation and inhibition, it also has reinforcement learning, can it actually explain the results that we see in the data? So this is the results of the model. This is one typical simulation. So first of all, we see that the network is able to increase the firing rate of a single neuron during learning. This is not very surprising because this is what we ask the networks to do, and we know that neural networks are very powerful mathematical entities. Whatever you ask them to do, they will probably do it, and if they don't do it, then we'll change the model until they do it. But the question is, what happens to the rest of the network? Do the unconditioned neurons evidence similar changes to the ones we saw in the data? So here are the changes in firing rate, and you see very nice large-scale changes that are symmetric between decreases and increases. And here is the changes in firing rate grouped by the correlation pattern with the conditioned neurons before the learning. And you see a very nice replication of the data. Negatively correlated neurons decrease the firing rate on average, while positively correlated neurons increase the firing rate on average. And actually, this makes a lot of sense when you think about the context of reinforcement learning. because in reinforcement learning, what we're doing is that we are reinforcing patterns of activity that lead to reward. And in our case, the reward depended on an increased firing of the conditioned neuron. So if we are looking at the correlation patterns, at the activity patterns of the pre-learning state, whenever the conditioned neuron increased its activity, that led to rewards, so positively correlated neurons will also increase their activity. And that increase in activity will be reinforced. And negatively correlated neurons, they will decrease their activity on average, and that decrease will be reinforced. So actually, this makes a lot of sense in the context of reinforcement learning, and it's very nice that we're able to show it here because we knew what is the important feature that leads to reward, in this case, the firing rate of the conditioned neuron. So these are the changes in correlation strength. Here you see correlation with the conditioned neuron. They became stronger, similar to the data. Here you see correlation between unconditioned neurons. They had a lot of changes, but they were relatively balanced between increases and decreases with a slight skew to the left. Again, very similar to the data. And when we looked at the pattern of functional connectivity changes, correlation changes between our different functional groups of units, between neurons that increased, decreased, or did not change the firing rate, we see that where the simulation was able to capture most of the changes. In asterisks are denoted those changes which were captured in simulation similar to the data, and you see that it captured 9 out of the 12 interactions that we saw. And in fact, these results and all the other results were very robust to the specific simulation. 2 minutes? I'm almost done. So all these results were very similar, were very robust across simulations. So we did 100 simulations with different initial conditions, different weights, different connectivity, and we got this result again and again and again. And I can also say that in those specific correlations, those specific interactions which were not replicated by the model were actually more than 97% of the time interaction in which the data did not significantly change. So maybe we didn't have enough data to see what the model could solve for us. Maybe in that case, the model was better than the data. So to summarize this work, we saw that volitional control of single neurons in primary motor cortex, it does not necessarily depend on activity and movement-related patterns. Rather, all our results, and we have actually more results I didn't have time to show to support this hypothesis, that activity patterns from the pre-learning state are being reinforced and are being adapted to accomplish the learning task. We saw that activity changes in firing rates and correlations following learning are relatively balanced, and this suggests the presence of a homeostatic mechanism for which we chose to model it as a balanced recurrent network. We saw the emergence of distinct functional groups that exhibited a very detailed and intricate pattern of changes in the functional connectivity, which gives us sort of an inside look of how the network learns. And we can predict to an extent how the activity changes in the network will occur based on the correlation patterns before learning. And this is an interesting result not only to support a reinforcement learning account of learning and a state-dependent account of learning, but also it can have some practical application. You can imagine that if we want to use neural control tasks in different ways, we want to elicit a specific activity pattern across the neuron, across the population, but we can only reinforce directly the activity of tens of neurons, then we need some way to predict what will happen to the rest of the neurons in the network, and this is one way to do it. And finally, we saw that a balanced recurrent network, a chaotic balanced network, which wasn't tweaked in any way, it wasn't finely tuned, because you can't really fine-tune a balanced network, it has too few parameters for that. And with a very simple reinforcement learning rule, can explain all the observed changes in the data. We have other data that it can also explain, which I didn't have time to show. And I have to say that I didn't know this when I approached Ran, but actually it's not trivial at all to do learning in the context of a chaotic balanced network. In fact, it hasn't been done before. And there was a key development by Ran which enabled this to work. So he will talk about it in further detail, so stay tuned for that. And I think that because balanced networks are so prominent in terms of explaining cortical dynamics, so the ability to use learning in the framework of balanced networks is going to be very significant. And actually, I think that this framework can be potentially extended to many different types of learning paradigms. So, well, I would like to acknowledge some people before you clap your hands. So first of all, I would like to acknowledge Eilon Vadia, my advisor and my mentor, for really teaching me about how to think about science. And he doesn't want to talk about him anymore, so I won't. Nofar, who helped with the experiments and also became my wife, so thank you for that. Other members of the Vadia Lab, Ran Darshan and David Hansel, for a great collaboration with regards to the computational modeling. Other members who help with the surgery and keeping the monkeys healthy, which is the most important part of the experiment. This work, my whole PhD, is dedicated to the memory of Dmitry Davidov, one of the most brilliant ELSC ICNC students passed away, and we remember him still. And thank you very much for your attention from the whole family.
Those of you who sit on the floor and want to take seats, it's now a good time if you like. We have time for one or two questions about maybe more about the data, and then later on we'll have more time. Yeah. Merav, and then David.
Do you know if you're recording from excitatory or inhibitory cells? Because that's
Yeah, we are mostly recording for excitatory cells. We are heavily biased toward the big layer 5 neurons, so these are the big bed cells which are excitatory.
So I'm curious about the difference between your results and Anderson's results. And I guess what my question is that sort of, I don't know, conditioning procedure for the animals see the targets coming on randomly being rewarded when, are they necessary in order to get the results? Another way to phrase that is if you don't do that, do you get Anderson's? Can you reproduce Anderson's?
Right, so we didn't have that experiment because I only had 2 monkeys, and I had a very limited time to do the experiment, so I didn't have time to do everything. So in order to be complete, I would have to have more monkeys and then use the same time of protocol that Anderson did to reproduce it. So I wasn't able to do it. But I think that the fact that we did find state dependence with a very similar state in which the learning starts, in which the overt behavior is very similar, I think that that indicates that the monkeys are using context dependence in that case. But again, we didn't have the resources to do the symmetric experiments and reproduce Anderson's result.
And we will need more larger monkey facilities to do this. Yeah.
You haven't said this explicitly, but I suppose that you did your correlation analysis based on the cross correlation, right?
Exactly. So I use cross correlations of firing rates in 100 millisecond bins. Yes.
If you refer to signal correlation, looking at just the firing rate, take advantage of the fact that initially I think you mapped the neurons' movements to.
Exactly. That's beautiful. So there was absolutely no relationship. That's a great point. There was no relationship between the movement parameters and the changes in firing rates due to learning. We found nothing. And I look at all different type of parameters: movement coding, preferred direction.
So no signal correlation.
Nothing. Absolutely nothing.
Neurons that have the similar
Nothing.
preference.
Right. So the first thing I thought was that neurons with the same preferred direction would actually increase the firing rate more with the conditioned neuron. Nothing. I found that, and that's why I said, well, it doesn't have anything to do with movements, because the preferred direction did not say anything. I did the velocity correlation, all type of grip, correlation with grip, nothing. We didn't find anything about that.
I should just comment that Ben worked about a year, and I was concerned that we will not have results because all this did not happen, and then he found a way around it. Okay. So I think we'll postpone all the rest for later. I just asked Nofar to show the result of Ben's study. He is right here.